All right. Oops. Okay, so today I'm going to talk to you all about the water quality monitoring that we've done in the last year in Rockport-Dorchester area. And my name's Julia Campbell. Um, can, oops. And I am EOS's watershed coordinator as of November 2020. Some of you may know Kelly Nicole, who was our former watershed coordinator, and she actually did all the monitoring at these sites. So I'm going to talk to you about the results today. So it's a bit of background about EOS Eco-Energy. We're a nonprofit in Sackville, New Brunswick. We serve the Tantramar to the Memramcook region and beyond, and we work to empower local solutions to reduce and adapt to climate change. And so, of course, with that, water quality is very important to us. So climate change can bring more extreme and more frequent weather events, which we have experienced here in Sackville, including flooding and drought. And Tantramar is one of the most vulnerable regions in New Brunswick, with us being such a low-lying coastal community and with our dike system here. Climate change can also magnify existing challenges and lead to new challenges, including increasing water temperatures, which can stress out our native fish populations and reduce their habitat. It has also led to increases and more frequent and intense algae blooms, including blue-green algae blooms, are also known as cyanobacterial blooms. And the photo of the three dogs there is from 2 or 3 summers ago in St. John, where there were dogs that had passed away from swimming in the St. John River. So algae is a concern in, with climate change. And there's also invasive species too that can become more competitive with climate change and outcompete our native species. So in 2017, EOS formed the Chignecto Watersheds Committee, and we started monitoring in 2018. And we decided to do this because there was no baseline data in our region, and we needed to better understand our watersheds and preserve and enhance our watersheds for the future generations. So just a quick recap of all the work that we've done at EOS. In 2018, we monitored 12 sites in the Tantramar River area and conducted riparian health assessments in the Tantramar River. In 2019, we ended up monitoring 12 sites in the Cape Tormentine and Peninsula area and conducted riparian health assessments in the Gaspereau River. And in 2020, what I'm going to talk to you about today, we ended up monitoring 12 sites in the Rockport-Dorchester area and performed habitat assessments in the Johnson Creek watershed. And all of these projects, there's final reports on them that can be found on our website in the watersheds project page. So these are the 12 sample sites in the Rockport-Dorchester area. And at each of these sites, we monitored field measurements using our HANA multimeter probe. And this consisted of taking water temperature, pH, dissolved oxygen, conductivity, salinity, and total dissolved solids. And we also took water samples at each of these sites, which were sent to RPC Lab, which is in Moncton, and they analyzed for upwards of 55 parameters that cover bacteria, nutrients, metals, and all sorts of other things. And we also measured chlorophyll A for the first time, which essentially represents plant and algal biomass in the water. And we ended up monitoring all of our sites from June to September this past season, and chlorophyll A was only monitored from June to August. So I'm going to jump into results for water temperature. Water temperature is affected by many factors, including the time of day that we sample, the depth of the water, and if there's any overhanging vegetation, such as trees that might provide some canopy cover and shade the stream. Water temperature affects many processes in the aquatic environment, and it directly influences who can live where, with different organisms having different tolerances of highs and low oxygen that they can tolerate or thrive in or breed in, and all that stuff. And so there is a guideline for streams. Streams should not exceed 20 degrees Celsius. So in June, we ended up seeing 6 sites exceed 20 degrees Celsius. And in July, we saw 2 sites exceed 20 degrees Celsius. And in June, we also saw 2 sites exceed 24 degrees, which under long term conditions can be lethal to salmonid species. And the reason for maybe why we saw the highest temperatures in June this year was because our hottest sampling day on average was in June. I was I was interested to see that the hottest day for our water temperatures was June compared to July and August. But as you can see in these graphs here, the yellow dot marks the day we took our sample, and the daily average air temperature for June was the hottest with 23 degrees. So moving on to dissolved oxygen. Dissolved oxygen is the amount of oxygen available in the water for aquatic life. And so how does oxygen get there? Well, it absorbs it from the air, it gets in from the movement of water, and also the photosynthesis of plants and algae. And the guideline for dissolved oxygen is that no site should be below 6.5 milligrams per liter. So for dissolved oxygen, we had 7 sites in June below this guideline of 6.5, 11 sites in July and August, and 5 sites in September. So we did see fairly low dissolved oxygen concentrations across our sites. And we also saw 6 instances of sites that were hypoxic, which is very low oxygen, and 2 instances of sites being anoxic, which is essentially no oxygen at that point. So a reason maybe why we saw such low oxygen conditions is because it was a very dry season. We received the least amount of precipitation in June, August, and September, the least amount in the last 7 years. So having received low amounts of precipitation means that our water levels were significantly lower this year. There was even some areas that had stagnant water, and so when water's not moving, you're less likely to get oxygen to replenish that water. And you'll notice throughout the following results that we did see higher concentrations, and a number of sites exceed certain guidelines too this year that maybe we wouldn't have seen in previous years if we had monitored where maybe we would have received more precipitation or water levels were higher. And I know that when talking to other monitoring sites in the province, other watershed groups, and even the lab that did the monitoring, they had said that this year, because of it being such a dry season, they saw much higher concentrations at a lot of sample sites. So moving on to E. coli. E. coli is an indicator of fecal contamination, which means that bacteria, viruses, and parasites could be present in the water. Many are harmless, but some can cause illness. And so there is a Canada Health guideline for recreational waters, which is a single sample maximum of 400 E. coli per 100 milliliters. So the results for E. coli are that 2 sites in June exceeded the guideline, 6 sites in July and August, and 3 sites in September. And you can see that Wood Creek and Tower Brook had exceptionally high measurements of E. coli. Now moving on to total phosphorus. Phosphorus is important for all living organisms, but when it is present in excess amounts, it can promote plant and algae growth, which of course can lead to a lot of other issues consisting of low oxygen and many other things. And so phosphorus can be found in high concentrations in certain rock formations. So it is important to know the geology of the sites that we're sampling, to not jump to conclusions about runoff and sewage, but it is found in it is typically comes from agricultural runoff and sewage. And so there is no specific guideline for total phosphorus, but there are what's called Canadian phosphorus trigger ranges, where eutrophic, that is a high amount of phosphorus, would have 35 micrograms per liter, and hypereutrophic would be above 100 micrograms per liter, which is very, very high total phosphorus. So we did see very high phosphorus across most of our sample sites. In June, there were 8 sites that were eutrophic to hypereutrophic. In July, there was 10. And in August and September, there were 11 out of 12 sites. And Peck's Cove Creek reached upwards of 700 micrograms per liter. So this site had very high total phosphorus. Now moving on to a slightly different way of displaying some of our results. Here we have a map of all the sample sites and an arrow pointing to all sites, where in this case, these are sites that were deemed as being brackish, which is a mix between a bridge between salt water and fresh water. And brackish sites have total dissolved solids between 1,000 to 10,000 milligrams per liter. So all the red arrows are pointing to sites that were deemed brackish, and next to the arrow are the months that these sites were brackish. All months would stand for June, July, August, and September, whereas Tower Brook, for example, was only brackish in September. So in June, July, and August, there were 3 sites that were brackish, and in September, there were 5. And these are the highest concentrations next to the arrows for each site, and all these concentrations are above guidelines. So you can see that Wood Creek had the highest concentration out of all sites with 31,700 milligrams per liter. So moving on to boron, the guideline for boron is 1,500 micrograms per liter. And in June, we had 1 site above the guideline. In July and August, there were 2. And in September, there were 3 sites above the guideline. And an interesting thing about boron is that all of these sites that are above guideline are tidally influenced sites. And boron is found in higher concentrations in like marine sediments and clay, and salt water. And so it, I guess, is not surprising that these tidally influenced sites that would have maybe some brackish water influence would have this higher concentrations of boron. And Wood Creek had the highest concentration with 4,300 micrograms per liter. So next, there were 2 sites that exceeded ammonia unionized guidelines of 0.019 milligrams per liter. There were 2 sites in August with the highest concentration being 0.65 milligrams per liter. A lot of sites exceeded the aluminum guideline of 100 micrograms per liter. There were 6 sites in June, 6 sites in July, 7 sites in June, and 8 in August and September. There was a total of 10 out of the 12 sites that exceeded these guidelines. And the highest concentration was at Penitentiary Road Creek with 950 micrograms per liter. We also had 11 out of 12 sites exceed iron guidelines of 300 micrograms per liter. There were 11 sites exceed guidelines in June, July, and September, and 10 sites in August, with Slacks Cove Creek down here being the only site that didn't exceed guidelines at least 1 time throughout the sample season. And the highest concentration was at Harvey Creek with 13,000 micrograms per liter. We also had 1 site exceed lead guidelines of 1 microgram per liter with 4 micrograms per liter. And lastly, we had 1 site exceed cadmium guidelines of 0.08 micrograms per liter with 0.2 micrograms per liter. So in conclusion for these monitoring results, it was a very dry season which could mean, which could be why maybe we saw a lot of these sites exceeding various guidelines. We haven't monitored these sites before, and so we're not able to tell like what's the range and the natural variability that these sites may display. But across monitoring groups in the province, it was noted that for people who routinely sampled, they did see a lot of sites above guidelines that typically weren't above guidelines. So a water sample is just a snapshot of water quality at that location at that point in time. So obviously, over time, we really hope to come back to these 12 sites to begin to learn more about water quality and understand any potential trends into the future, and tracking changes that are occurring in our watershed. So I'm going to jump into some of our habitat assessment results in the Johnson Creek watershed. And habitat assessments consist of taking stream channel conditions, riparian zone, and aquatic habitat conditions. And each of these assessments that assess these various conditions are taken at every 100 meters throughout the stream. And we ended up using methods laid out by the Kennebecasis Watershed Restoration Committee. So we ended up doing a total of 57 assessments, which spanned 5.7 kilometers throughout Northbrook, Johnson Creek, and Eastbrook. And for each of these assessments, we ended up using a point system to assess each of these sites. And so the point system uses factors that define a potential restoration site to assign sites points. So if sites had dissolved oxygen under 5 milligrams per liter, they got a point. If sites had heavy erosion over 25% on the stream banks, they got 4 points. And all of these points were added up to either define sites, to rank sites as either healthy with 0 to 3 points, healthy with problems, or unhealthy with upwards of 11 points. So our results were that 73% of the assessments were healthy, 25% were healthy with problems, and 2% were unhealthy. So some of the factors in these sites that were either healthy with problems or unhealthy, or even sites that had points in general, was that there was a large amount of grassy vegetation. And grass has a shallow root system, which makes these stream banks more prone to erosion. And it also has low shade cover. So if you have a lot of grass, you're not going to have a lot of trees and shrubs that would have that nice canopy cover that shades the stream. And with a stream like this that doesn't have a lot of shade, it's more prone to higher water temperatures that can stress out fish and other organisms in the habitat. Sites also displayed a lot of fine sediment, and fine sediment can suffocate aquatic insects, which are really important food for bugs and other organisms, or for fish and other organisms, and more bugs too. It can decrease water clarity, which can stress out fish, and shade plant and algae growth. And excess plant and algae growth obviously isn't a good thing, but obviously plant and algae is needed in the system. It's the base of the food web, and it provides habitat and food for fish and other organisms, and so it is very important. And all this excess fine sediment can also degrade fish habitat because certain fish species prefer these rocky stream bottoms for them to lay their eggs, and all this fine sediment that can easily be picked up and resuspended can stress out fish and get into their gills, and it's not very healthy. So what are some recommendations? Well, obviously, I have a limited amount of experience in different watersheds, and so the recommendations that I make are just some of the things that I could think of with my experience, but further investigation for any of these sites that may be healthy with problems or unhealthy is needed to decide what's best for these sites. But some of the recommendations that I made could consist of something as easy as planting native trees or shrubs in sites that have high amounts of grassy vegetation to strengthen the stream banks with plants that have deeper root systems, provide some more shade, and all that kind of stuff. So for sites that have a high amount of fine sediment, we could also remove large debris that's in the stream to help free up some of the sediment and flush it out of the watershed and restore some of those rocky stream beds. We can also install what's called brush mats, where what I've done in the past is you take spruce boughs, so take the boughs off of some low-lying spruce trees, and you lay them down in a mat. I know the Kennebecasis watershed also uses willow and sticks them into the bank to reinforce the bank too. So if you were to use spruce boughs, you lay them down, you put a bunch of stakes in and tie it down with some twine. And over time, fine sediment is going to pass over this mat and it's going to collect and form this nice naturally sloping stream bank where sediment builds up and it removes it out of the stream. And over time, some nice vegetation from the stream bank can grow over top of it. So again, these are just some suggestions that I had, but further investigation would be needed for the specific sites that we would pick if we were to do any restoration activities. So what do we want to do in the future? Well, currently we're waiting to hear back on funding. So we really hope to return to the Tantramar River watershed where we monitored 12 sites in 2018. And we would also really like to do habitat assessments in Joe Brook, which drains into Silver Lake, which is a really important area for recreational swimming and fishing and all sorts of activities. And Joe Brook's also a really popular site for angling. So with that, I would love to open it up to any questions and comments from anyone. And if you also wanted to email any comments or questions, my email is at the bottom. And of course, there's always all the final reports on our website if you wanted to check those out as well. So yeah, thank you everyone. I don't know if you're able to unmute yourself or in a chat box anywhere, ask any questions, then feel free.
Yeah, sure. Well, thank you so much for your presentation. That was really informative. I guess my question is, you said that in some of the streams there was high E. coli. What are some of the health impacts of that?
Well, I don't specifically know the specific health impacts, just that people can get sick. Like when you think Parley Beach and such, around Shediac area, it's a big concern for public health. So not all E. coli, like the presence is bad, but obviously too much of it, then we're concerned about people's health and such. I don't know the specifics, but there are guidelines for a reason laid out by the government.
Right. And would that be mostly caused by like having cows nearby?
Yeah, or even if people's properties have sewage that's leaking from old septic tanks, or yeah, any runoff from any agriculture like livestock, or even like certain fertilizers or manure that people would put on the property as well.
Interesting. Well, thank you.
Thanks, Catherine. Yeah, Vasant.
Hi, Julia. Thank you so much for the presentation. It was like very interesting to see the elements. My question to you is like, I saw that like in one of the first slide, you told that there was no baseline data.
Yeah.
So I was kind of curious to know without baseline data, like how can we, what sort of like the data or thoughts you took in terms of like deciding, okay, this is the extent things have gone worse, or this is the extent things have become better. You know what I mean?
Yeah, so obviously with like, it's excellent that we took this first year of information and now we have all this data. It's also uploaded on Atlantic Data Stream, which is a portal that a lot of groups in Atlantic Canada use to upload their data. So with moving forward now, we have some of this baseline information where when we intend to continue monitoring in the future, we can kind of detect any potential trends in the area, or if, for example, there was something that happened, and there was damage to the watershed, then we have this year of data that we can compare. Maybe this year isn't the most like natural year. Obviously, we are experiencing changes in our watershed, but it's better than nothing. And it's great to have this data, and we hope to collect more in the future to be able to actually compare what are some changes that are going on and such. So I don't know if that really answers your question, but
Yeah, it kind of does, because I was like, well, like like I was wondering that if we have some resources to go back to and compare, how things have changed, and that'll be like a good start, right? So
mhm.
I think, like you said, if this is the way things can like go forward, like you creating these data and then setting a baseline, and maybe 5 years, 6 years from now, you can go back and compare how things have improved. So yeah, and also you told me that like there's also limited data available in terms of like chemicals that has been like iron or ammonia or phosphorus. You don't have like exact standards or that's been laid out so as to like this is the amount that should be present, things like that. So yeah, I believe like if there are standards put down properly, then that will be even more helpful for the research, I guess.
Yeah, for sure. Yeah, and the data is always like publicly available if anyone someday decides to pull it up and they want to do more research on it as well, which is great.
Thank you for that. Thank you.
Thank you. Samantha?
Hi, sorry, trying to figure out Zoom. Thank you so much for this presentation. It was really interesting. I was just wondering if you could touch a little bit more on what RPC kind of contributed to this, because you said they touched on 55 parameters. Could you just give me a little bit of a broader view of what was done?
So in our final report, there's tables full of all this information. So they would have measured for like all different types of metals, and like done more measurements on like conductivity, and they did all the phosphorus and E. coli. There's all sorts of other stuff. I can't think of it off the top of my head. But in our final reports, all of it is there. But yeah, everything that I talked today, except for like temperature and some of the field measurements, was all done by RPC.
Awesome, thank you so much.
Yeah, Roland here. I have a question. I wonder if when the habitat assessments were done, was there any information gathered or some notes taken on how well the legal buffers are intact along the streams?
To my knowledge, I don't think any notes like that were taken. I know there was a lot of logging activity in that area in the like late 1900s, I believe. And a lot of it is regrowing. So I don't know how recent there's been any activity within like the areas that we did the habitat assessment. But I know throughout previous work that Kelly Nicole had done, there were photos and stuff taken to kind of record some of that, because obviously those areas would be really great for any future potential restoration projects or partnering with local landowners to restore some of those stream banks and such. So, but there is an interactive map that captures photos. They have a bunch of photos for every assessment that was done, and the raw data that was taken was transcribed onto Google Docs. And I forgot to show it. How do I escape this? I'm gonna zip back quickly. Oops. This photo here. So this is an interactive map that everyone can go check out. It's linked on our website. and in the habitat assessment final report. And if you click on any of these points, you'll pull up a Google Docs link and you can click on it and find any notes that they would have taken at each of the assessments. And if you click on the photos, you can flip through a bunch of pictures. So I don't think there's like specific information on the buffers and stuff like that, but there would be lots of useful notes in there. And it's obviously something that we want to continue to do with recording all this information when we want to plan future monitoring or potential restoration, because EOS does cover such a large area. So there's lots of work to be done.
Super, thank you.
Thank you. Are there any more questions or comments? All right, well, if you guys have any other questions, feel free to email me, reach out, check out the reports on our website if you want to see more. And thanks so much for attending tonight. I really appreciate it, talking to an audience and you guys answering and asking questions and such. So I hope you all have a good night and take care.
Thanks, Julia. Great presentation.
Thanks, Brittany. Bye.
Thank you.
Thanks, Julia.
Bye.
Thank you.
Thanks, Julia.
Thank you.
Thanks, Julia. Have a great evening.
Thanks, Miranda. You too.
Thanks, Julia.
Thanks, Perry. Take care.
You too. You did a great job.
Thank you.
Have a good night.
Thanks, you too.
Thank you.